Hello, everyone. I'd like to welcome you today to the webinar focused on plant endophytes and phosphate solubilizing bacteria. My name is Scott Lohman. I'm Director of Research here, and before I introduce our speaker, I'd like to give you a little background on the Institute and our research department. The Institute was created in 2003 as a mechanism of economic transformation in Southern Virginia. We do that through 5 different divisions. One is advanced learning, where we house over 200 high school students during the day. Number 2 is advanced manufacturing, where we train students with advanced skills and techniques for 21st century manufacturing and workforce development. We also have economic development in-house, as well as conference services. Our research department has historically been focused on research, although we do chemistry and others, and mainly we support industry. Because the Institute is a higher education center in the Commonwealth of Virginia, we do receive state funding, and that makes us different to work with than most academia and other industry. Because of that funding, we're able to go into new areas, such as plant endophytes and robotics. Our research also includes analytical chemistry, and mainly we focus on contract research. We do IP co-development, we support industry development and new industry in our area, and we're even diving into workforce development. But with that, I will go ahead and turn it over to our esteemed colleague, Dr. Chuanxing Mei, and let him take it over from here.
Thank you, Scott. Good afternoon, everyone. Thank you so much for joining us in this webinar. Before we talk, I want to briefly introduce myself. I'm a chief scientist here. I got my PhD in the University of Illinois, Urbana-Champaign. And after that, I went to Global Foundation for my postdoc training. After that, I work in University of Arkansas, Michigan State University. In 2007, I joined the Institute for Advanced Learning Research. In the beginning, I worked on bioenergy crop switchgrass miscanthus using beneficial bacterial endophyte for low-input sustainable feedstock production. Recent years, we built up bacterial endophyte library for applied research. Let's start today's webinar. Today, I will talk about the beneficial bacterial endophyte for phosphate solubilization. Before my talk, I want to introduce our team. Dr. Scott Lohman, Director of our research. He just introduced our institute. Sajiva is a plant pathology guy. He is responsible for biocontrol agent screening. Robert Kitchen is molecular biology. He is responsible for characterization of bacterial endophyte and also lab greenhouse work. Amy Turner, he is responsible for tissue culture, and also he can do everything in a lab and greenhouse. Jack Hu, he is an analytic chemist. Amy Zhu is joining us 1 year ago. He is doing hydroponic work in greenhouse. Here's my outline for my talk. First, I will briefly talk about the bacterial endophyte library, and then how we to characterize our bacterial library, how to use our bacterial library. And then I will focus on phosphate solubilization bacterial endophyte. The final, we have Q&A. So please submit your question through Q&A box. 2 years ago, we are launched the Plant Endophyte Research Center. We're focused on bacterial endophyte. Here's how we established the bacterial endophyte. So we connect the plants from various environmental conditions, and then do a surface sterilization, isolate bacterial endophyte. We create the stock. And then, so right now we have 2,000 bacteria strain. How we can do this with 2,000 bacteria strain? So we do all kinds of screening. So here's part of work that's for biocontrol agent. Sajeva, in last webinar, he presented this work. If you missed, so you can go to our website to listen to his record. Another big part is plant growth promotion and plant nutrient acquisition activity. So here we do a ACC deaminase assay, IA quantification, nutrient nitrogen fixation. So today I will talk about the more detail for phosphorus solubilization. So after we do all kinds of characterization, so we know which bacteria has what kind of function. So we do some in vitro plant growth promotion test. Here we use Tofesco, this grass, for our routine screening for plant growth promotion in vitro. So you can see here, so we get the really good results for plant growth promotion. Also, you can see picture, that's totally different. This is in vitro work. However, in a greenhouse field, there's much difference. So we also have new technology. It's called smart image. So we can detect the plants in real time. So here's a image. You can, based on image size, you can see plant growth difference. We do lab, greenhouse, and a field study. Here's a greenhouse study for strawberry. Here's field work for strawberry, field trial. Next, I will focus on phosphorus solubilization bacteria. Here's the situation. Only 0.1% total phosphates in soil is available to plants because most are insoluble. Plants require 30 micromole of phosphate for maximum production, but only above the 1 micromole is available in many soil. So here's some insoluble phosphate. So calcium phosphate, aluminum phosphate, ferric phosphate, also some organic phosphate is phytate and other organic matter is plant residue. So here's a dilemma. Total phosphate amount is abundant in soil, but soluble phosphate is limited to plant. So this figure shows you how phosphates cycling in soil. So here's the input. Farmer apply apply fertilizer, plant residue, and other stuff. And some become, insoluble, fixed by, other calcium, ferric, aluminum, and some plant use is about 20 to 30% plant can use this fertilizer. Other will run off and contaminate water. Here's some is also called eutrophication. So eutrophication, cause big problem. Cause dead zone. So here's the Ox 30 this year report. This Gulf of Mexico, the dead zone is more than 6,000 square mile. That's big. So another example is toxic algae booming, which left 500,000 people without drinking water in Ohio. So here's how we do research. The common use the two medium. One is PVK is actually this Picovesicia. Another is NBRIP, National Botanical Research Institute, phosphate. This is both from India. So another version is enhanced NBRIP. The difference is just use double glucose and double magnesium chloride. This is use 20 gram glucose per liter, use 10 gram magnesium chloride. So if you just screening, you don't need, based on cause effective, you just use the NBRIP. Here's the solid medium, shows bacteria grew and then solubilized insoluble phosphate. We use calcium phosphate, so it's the medium chloride. And after bacteria grew, you can clearly see halo. This is bacteria no solubilization ability. Also, NBRIP is much better in broad, means in a liquid culture. It's about 3 times higher than PVK. So right now we just detect soluble phosphate in medium, use Murphy and Rani 1960 methods to quantification. So after that we got some really high solubilization bacteria. Here's a table show you we pick up 5 representing different species. So we pick up 2 from Pentoria. One is Pentoria vigorus, another Pentoria agrolomis, and one from Pseudomonas, another 2 from Bacillus. So here's gene bank accession number. So you can find this sequence in gene. Here's the tissue we isolate from, and this is plants. We also did a gram stain. Based on 16S sequence, we constructed this phylogenetic tree. So this board fan represent our bacteria. So you can find, this the same species, But actually, because bacteria change so much, usually, one species can have 1,000 or 100, at least 100 some different, strings. And also their function is different. This table shows you we use different insoluble phosphate compound to see how much the bacteria can solubilize phosphate. This first one is calcium phosphate. We have really high number. And the second one is called hydroxypeptide. This also is good, is around 100. But another one, aluminum phosphate, ferric phosphate, they have minimum. In addition to phosphosolubilization, these bacteria also have other plant growth promoting traits. So you can see we measure IA, nitrogen fixation, ACC deaminase, siderophone. This 611 string have all four, and some of this 1035 only have one siderophone. So that means one bacteria not just have one trait for solubilization, also have other, plant growth hormone, nitrogen fixing, or ACC deaminase. Here's the experiment. We test different insoluble phosphate compound. The one is calcium phosphate, ferric phosphate, hydroxypeptide. So you can see this string 1325 is give best results. Is almost 4 times high than control. Here's to show you some picture how plant grew in a medium. So this is 1325. Here's in vitro work. However, in a greenhouse, this situation is different. This you cannot get 100% increase. This only get maybe 20% increase. Here's we use pepper and tomato. So you still can see in a pepper, you can see 3025 and 632, 1033. Significantly increase plant growth compared control. You can see different A, B, and in a tomato, also 1325 and 632 have significantly promoted plant growth. So based on our results, 3025 is best. So we also want to check this bacteria can, hydrolyze organic phosphate. So acid phosphatase and phytase, this phosphatase enzyme hydrolyzes this ester phosphate bonds, releasing phosphate ion. And the phytase enzyme hydrolyzes phytic acid. So here's to show you a phytic acid. This one molecule has 6 phosphate molecule. So this is major storage in a field for organic phosphate. So here's the enzyme assay. So you can see all bacteria has kind of activity. However, this 632, they have highest acid phosphatase, 700. Next, we want to know what kind of mechanism bacteria can solubilize phosphate. So one common mechanism is bacteria produces organic acid, and then reduce pH, so solubilize this insoluble phosphate. Here's the assay. We use HPLC to detect the gluconic acid. So you can see is high levels gluconic acid in bacteria medium. And here's the pH change. So from 6 something to 5 something. Also, we find the pH and gluconic level has a negative relationship. This R squared is 0.82. So this show you a mechanism of bacterial endophyte to soluble in organic or organic phosphate. So here's acid production, and here's show you a calcium phosphate and acidification. So you release this phosphate, and chelating, you also release this phosphate. Here's also bacteria produce siderophone. Siderophone has a high affinity to ion, so also release phosphate. In organic phosphate, there's 2 enzymes and release some phosphate ion. Based on our results, we also supported this organic acid mechanism. Because this bacteria has glucose dehydrogenase, which directly oxidizes glucose to gluconic acid. And in this genomic sequence, we also find another PQQ. This a cofactor for this enzyme. So application of phosphate solubilization bacteria is eco-friendly, economically sound approach to overcome the phosphorus dilemma in soil. Also definitely increase phosphorus use efficiency. People also report bacterial consortium maybe this most effective in solubilization insoluble inorganic phosphate compound and mineralized organic phosphate. So we still have more work to do in this area. However, consistent results from lab, greenhouse to field trial is required for success application. Also, micro fermentation and formulation for consistent high quality is required. Last, I want to introduce our new initiative. Virginia Tech and RIA Controlled Environment Agriculture Innovation Center, which features indoor ag, greenhouse hydroponic, aquaculture, and aquaponic. Here's Dr. Michael Evans, Director of School of Plant and Environment Sciences, Virginia Tech. He's a really expert in this area. We're also involved in hydroponic work. Here's to show you a beneficial bacterial endophyte increase hydroponic lettuce growth. Dr. Zhu just presented these results in American Horticulture Associate annual meeting in Denver, Colorado. Thank you for your time.
Thanks so much, Dr. May. Excellent presentation. If everyone would submit their questions through our Q&A, we'll be happy to take those. We had a couple submitted before as people were signing up. And one question was, are these microbes compatible with fertilizers and or other chemical products? And I'll turn that over to Dr. May to answer that one.
Yeah, this is a good question, I saw one publication, they suggest the use phosphate solubilization bacteria with rock phosphate, because rock phosphate is a slowly released phosphate. Yeah, we don't have any results experiment for this area. So for chemical fertilizer, phosphate, so this is already in a soluble, so maybe not a good idea for combine with phosphate solubilization bacteria.
Okay, thank you. Does anyone else have any questions? And while we're waiting, I'll go ahead and let you know that we are planning to do a couple more of these. We'll focus on ACC deaminase, maybe some hydroponic work. Any suggestions would be appreciated. We may also have a webinar that updates and highlights our new smart table system. That's our precision imaging system and what it can show. And otherwise, I don't believe we have any other questions. So with that, I would just want to thank everyone again for tuning in today and let you know that if you are interested in any of our bacteria or working with us or contract research or anything along those lines, please reach out and contact us through the email that Dr. May presented as well as through our website. Thank you so much and hope to see you again next time.